So today I have a little bit cold infection, some throat infection is there.
I see.
Yeah. So my voice is so far good, right? I can.
I think
Everybody can hear, right?
if you had not mentioned that, we would not have realized.
Oh, okay, okay.
Hello, good evening, Dr. Vinu.
Good evening, sir.
Myself, Vijay.
Oh, Vijay.
Yeah, good evening, Manoj sir. Hello.
Yeah, good evening, Vijay.
Good evening, Ram sir. Hello.
Good evening. Good evening.
So we have a YouTube podcast of this lecture. So just the link is actually sent to the chat box. So we want to forward it to somebody who may have an interest in attending this, please.
In fact, I have forwarded to all the TSI group, and also probably Dr. Prem Nair will be joining.
Yeah.
So we are expecting Dr. Prem to be joining.
Oh, Dr. Prem is joining?
It's likely because I've sent the link and Dr. showed some interest, and they are doing the project also. They have 5-6 projects.
Very promising area, actually. You know?
Yeah.
A lot of opportunity in Kerala or in India to set up industries in this area.
Yeah. Especially in the present scenario, yeah, there's so much of opportunity.
Yeah, right. It's 7 o'clock sharp. I think we can start.
Yeah, yes, sir.
So shall we start, sir?
Yes, please.
Okay. An investment in knowledge pays the best interest, says Benjamin Franklin. A warm good evening to one and all present here in this virtual gathering. Most respected guest of honor and our eminent speaker Dr. Vinu Mohan AM, esteemed dignitaries, delegates, and my dear participants, I cordially welcome you all to the third distinguished lecture series jointly organized by IEEE IEEE Kerala Chapter, Telemedicine Society of India Kerala Chapter, and Digital Health India Association. Today's session focuses on the topic Emerging Trends in Wearable Electrochemical Sensors. Without further delay, let me now invite Dr. Manoj PS, Chairman of IEEE IEEE Kerala Chapter, to welcome the virtual gathering. Sir, please.
Yeah. Good evening. I'm very glad to be here and extend a warm welcome to Dr. Vinu Mohan AM, who has kindly agreed to deliver a distinguished lecture on this topic, a very interesting topic of electrochemical sensors. He has been a faculty, DST inspired faculty at Electronics and Electrocatalysis Division. Really glad that he is able to deliver this lecture. Welcome, Dr. Vinu Mohan. Second, I would like to welcome Dr. Prem Nair, who has been organizing several such events jointly between IEEE EMBS, Telemedicine Society of India, Kerala Chapter, and Digital Health India Association. Welcome, Dr. Prem Nair. Third, I would like to welcome Dr. Ramkumar Menon. He's a neurosurgeon, also an entrepreneur. He's also a biomedical engineer and a multifaceted personality. Welcome, Dr. Ramkumar Menon.
Thank you, sir.
I would also like to welcome Dr. Uma Nambiar, Digital Health India Association, who has been another strong collaborator of IEEE EMBS Kerala chapter. I would like to extend a warm welcome to my IEEE EMBS colleagues, IEEE members here, TSA Kerala chapter members, DHA members, as well as the student volunteers here and distinguished audience present here in this time. A very warm welcome to one and all. Thank you.
Thank you, sir. I now invite Dr. Ramkumar Menon, Executive Member of the Telemedicine Society of India Kerala chapter, to introduce our most esteemed speaker. Sir.
Thank you. Good evening and welcome to the distinguished faculty. And let me thank each and every one of you who are here to hear Dr. Vinu Mohan. I've been given the privilege to introduce Dr. Vinu Mohan AM. So Dr. Vinu Mohan AM, who is currently a DST Inspire faculty of the Electrodics and Electrocatalytics division at CSIR, Karaikudi, Tamil Nadu. He has a significant area of research interest, and one of that is actually wearable microfluidic sensors for multiplex non-invasive monitoring of biomarkers from such sample. That's a very, very kind of an interesting topic, considering that it's a no-touch, no-go technique nowadays, wherein you minimize your contact with the patient. And it seems to be very interesting. I think we are looking forward to a gala talk. And his area of interest also includes briefly introducing his academic proficiency. As I mentioned, he is currently working as an Inspire Faculty Fellow DST at CSIR Karaikudi, and he's been working there since April 17. California, San Diego, US, for a period from 2015 to 17. Holds his PhD in chemistry from the National Institute of Technology, NIT Trichinapalli, and completed his master's in chemistry from Christian College, Katakada, that's Trivandrum, Kerala. So I think there's a lot of academic activities and a lot of research activities going on with Dr. Vinu Mohan. Doctor, we welcome you to this meeting.
Yes, thank you, sir.
Thank you very much.
Thank you, sir. Without holding up the session, let us all synchronize our attention to our illustrious speaker to hear his insights on emerging trends in wearable electrochemical sensors. Once again, welcome and over to you, sir.
So, good evening, Onandal present here. Thank you for your nice introduction. So, myself, Dr. Vinu Mohan, working as DST Inspire faculty at CSIR Sikri Karaikudi. So today, now I'm going to elaborate about emerging trends in wearable electrochemical sensors. So, does the slides are moving?
Yes, yes. We are seeing the contents right now.
Yeah, yes. So now I'm going to elaborate initially the current clinical analysis, especially for disease diagnosis, how the clinical practices are going on. Then what is the importance of point of care testing devices for clinical analysis. Then I will move on to wearable electrochemical sensors. So why it is important, what are the different types of electrochemical sensors, and the properties of electrochemical sensors are really important. It should be flexible and stretchable and self-healing in nature. And using these wearable sensors, we can detect biomarker of interest from different biofluids like sweat, saliva, and interstitial fluid. So I will discuss how we can fabricate these flexible sensors for this biomarker application. Also, stretchable and self-healing properties of the sensors will be elaborated. So currently, if one person is feeling not good, so what we will do? So we will go to the hospital and we will consult a physician. So the physician, they will initially give some medicines, and again, if the symptoms persist, again you will go to the hospital, and the doctor will say for some blood analysis. So we can have not only blood analysis, other types of some biofluids are available from our human body. They are blood plasma, urine, saliva, sweat, and cerebrospinal fluid. So it depends on our the symptoms, the disease symptoms. Doctor prescribe different analysis. So, but the main problem associated with this clinical analysis is it is a time-consuming process. Based on sometimes some labs are very rush, and we have to give the blood samples and wait for some hours. But if it is the symptoms are under control, then no problem. But if the person is in an emergency situation, but then these hours matters, and sometimes this delay leads to the death of the person. So usually people are using in the laboratories are using bulkier instruments, and they are using colorimetric principle, not electrochemical, not exactly electrochemical principle. Very few of the current clinical analysis are based on electrochemical analysis. Otherwise, it is optical sensors. So basically, they measure the absorbance, which is proportional to the concentration of the analyte present in the biofluids. So we can detect metabolites, electrolytes, proteins, and drugs. So these are some of the examples. We can have a blood analysis, and we can detect the concentration of sodium, potassium, chloride, urea, creatinine. So these are all the biomarkers present in our human blood samples. So these some of them are especially using for clinical purpose. Some of them are for a fitness monitoring application. So hemoglobin, glycated hemoglobin, uric acid, cholesterol, lactate. These all are really important for analyzing a person's health status or physiological status. So, as I said, the current clinical analysis sometimes it leads to the delayed treatment because of the time delay in blood analysis. So nowadays in the market, several point of care testing devices are available. So as a definition, POCT devices are the diagnostic testing devices, and we can have the analysis at or near the patient site. So no need to depend on clinical laboratory or trained healthcare personnel. The patient itself, they can have the analysis. They can take one example is the glucometer available in the market. We can prick our finger and get the blood samples and do the analysis. So using this POCT devices, we can improve the medical outcomes, and we can improve the economic outcomes, and decrease the therapeutic turnaround time. That is the time required for test request to patient treatment. So POCT devices have several advantages. We can get the data very quickly, and we can have the analysis directly from the blood sample or from the urine sample. And no need of any pre-treatment procedures. Sometimes in blood analysis, they do some pre-treatment analysis. But using these POCT devices, we can test the biomarkers directly from the biofluids. So there are several types of POCT devices. One type is using for primary care analysis, and another one using for secondary or tertiary care analysis. Primary care means we can have the analysis at home, or at a community pharmacist, or at a health centers, or workplace clinic, or a physician's office, or even at a paramedical support vehicle. But in the case of secondary or tertiary care, that will conduct by not by the patient itself, by the any supporting staff or a doctor. especially in the emergency room, admission, intensive care unit, or in the operation theater, or in outpatient clinics, or in the wards. So mainly Vivocity devices can be divided into three categories. The one is the strip-based analysis. Basically, it is more qualitative analysis. We can get the information like a yes or no. One very good example is a pregnancy test strip. So we can introduce the sample, and within a few seconds we will get the qualitative information, or sometimes we will get semi-quantitative information. And in the another one, the strip or the cartridge integrated with a reader. One example is a glucometer. Nowadays, it's widely available in the market. The third one is the multiple use quantitative cartridge, which needs to be integrated with the benchtop devices, not a simple meter. Sometimes we need some pre-concentration or some analysis before some treatment before the actual analysis. So for the multiple use cartridge, we can detect several electrolytes, metabolites, bilirubin, cardiac marker, or a C-reactive protein. So these are some of the examples of Vivocity devices currently available in the market. They are some of them are handheld meters. We can able to carry wherever we are traveling. Some of them are portable in nature, and some needs to be integrated with the benchtop. And we can have ex vivo analysis sometimes continuously, like overnight we can have the analysis. And nowadays the advances in Internet of Things, it gives the facility to save the data, and like over a period of say for example 1 week we can have the analysis, and directly we can send the data to the physician, and we can avoid frequent visit to the physician or the hospital. So I'm not going into the details. So before going to the wearable electrochemical sensor, I'm going to detail some information about the how electrochemical sensors are working. So in electrochemistry, we need three electrodes. One is a working electrode, then counter electrode, and reference electrode. So we need to introduce these three electrodes in the biofluids, and then we have to connect these electrodes to a potential stand for further analysis. So basically, we are applying some potential and measuring the current response from the solution. Sometimes when we apply the potential, the molecule or the biomarker present in the biofluid, it undergo oxidation. So when oxidation happens, it produces some current response. Sometimes it undergo reduction. That times it consumes electrons. So this current flow, either oxidation or reduction, that will be that response will be proportional to the analyte of interest, the analyte concentration. So these are the electrochemical cell we are using in our lab level. So for all basic characteristics, we are using a small glass vessels, and we are using like a disc type of or a rod type of electrodes. We are introducing these three electrodes in a cell, which have some supporting electrolyte plus the analyte of interest, and then we used to do the analysis. But for practical application, we cannot have this ideal setup. This glass vessel, everything is very difficult. So for that purpose, we need to have these three electrodes in a same platform. So we can use like some printing technology or any advanced fabrication or coating technology, and we can coat these three electrodes on a same platform. So nowadays, screen printed electrodes, we all know, they are widely using. In our glucometers, we are using glucose strips. So all these, majority of these glucose strips are based on screen printing technology. So here you can see a screen printed electrodes, which consists of a working electrode, a center part is working electrode, and this one is the counter electrode and a small reference electrode. And from these electrodes, there is a silver-based contact. Silver, we can use both. Silver means AgCl. So it can function as both a contact as well as the reference electrode. And carbon, of course, carbon-based inks are very cheap and inexpensive. So and the over potential for carbon is really less compared to other electrodes. Easily we can functionalize carbon electrodes and use. So nowadays we can have these like portable analyzers, like a handheld analyzers are available. Some are like a pen type of analyzers are available. And these analyzers consist of some Bluetooth chip, and we can have our collected information, the analyte of interest, the quantitative data, we can transfer into the mobile phone, and whenever we are free, we can access those data. So, especially for wearable application, wearable electrochemical sensing application, we are using these two techniques, amperometric detection, and another one is potentiometric detection. The reason is we can easily prepare the controlled electronics. So these screen printed electrodes, we need to connect with some PCB circuit board. So in the case of amperometric detection, it's very simple. We have to apply some constant potential and measure the current response. In the case of potentiometric detection, no need to apply any potential, but we have to measure the open circuit potential at zero current. So these two techniques, especially amperometric detection, we can use for all enzyme-based sensors. Why we need an enzyme? So enzyme we are using for getting selectivity. So we talk about these different biofluids like human serum or plasma or saliva. All these biofluids are very complex in nature. So it consists of not one biomarker. It consists of several biomarkers, and many of them are electrochemically active. If it is electrochemically not active, no problem. It won't give any interference. But majority of the biomarkers like glucose, lactate, uric acid, ascorbic acid, dopamine, all these biomarkers are electrochemically active. And when we apply a particular potential, if it is little bit high, like a 0.3 or 0.4, that itself high potential. So at that time, all the other analytes or the interference, everything it will undergo oxidation or reduction, and you cannot have the selective detection. So in order to improve the selectivity, we have to functionalize with the specific moieties like an enzyme. So for each analytes, we have separate specific enzymes. For glucose, it is glucose oxidase. For lactate, is lactate oxidase, or like a dehydrogenase-based enzymes are available. So this is the enzymatic mechanism. One example from glucose. If you are using glucose oxidase, in the presence of oxygen, it undergo oxidation and converted into gluconic acid, and H2O2 will produce as a side product. Then H2O2 is further electrochemically active. Sometimes we can oxidize H2O2 or reduce H2O2. So basically here we are initially oxidizing glucose with the help of an enzyme, and then we are measuring the side product, the H2O2. So in that case, we will get a much more selectivity. So the formed H2O2 will be proportional to the concentration of the analyte, that means glucose. So we can have, because we are directly oxidizing glucose, we have to apply high potential. So at that potential, say for example, above 0.6 volt for carbon electrode. So at that potential, all of these interference I mentioned earlier, it will undergo oxidation. But when we oxidize through enzymatic method, then we can oxidize this H2O2 at a low applied potential. So coming to the wearable sensor. So nowadays, the global market of wearable sensor is particularly focused on physical, like a monitoring of physiological parameters, like heart rate, oxygen level, then muscle movements, ECG, body temperature. So these parameters. But some few percentage is only for wearable chemical sensors. Okay. So there are three categories. One is clinical diagnosis. We are using chemical sensors. Then for fitness monitoring application, we are using all physiological sensors. So then for the third one is rehabilitation. For clinical diagnosis, one product is commercially available in the market. This is the wearable glucometer. So which is one of the electrochemical sensor. So it consists of, so here you can see the person is, the person own a small patch, that is a glucose patch. So that patch consists of some micro needles. So when we attach these sensors on the epidermis, what will happen? It is like a semi-invasive analysis. It penetrates the upper part of the skin, that is the epidermis, and which can access the interstitial fluid. So the interstitial fluid is the fluid surrounds the cells in our human body. If you are seeing the cell, on the cell, outer exterior area of the cells are covered with these extra cellular fluid. So from the blood through capillary action, these interstitial fluids, which consists of several analytes like lactate, glucose. So several biomarkers are present in the interstitial fluid also. But the concentration is not exactly similar to the blood, but they are proportional to the blood. For example, if the glucose concentration is 1 millimolar, we can't expect the same concentration, the same level of glucose in interstitial fluid, but which is proportional, like 100 times less than that of blood glucose level. So here I can see the human skin. The upper part is called epidermis. So these are the cells, and I can see the interstitial fluid around the cells. So this is the electrochemical sensor glucometer attached on it, and we can access the glucose and have the analysis. And not only to this meter, if we can download some app from the company's website, and we can use in our smartphone itself. So just open the smartphone, and with a single click, we can have the glucose analysis. So the second category is for physiological monitoring. I said there are several companies are currently available, like Fitbit, Samsung, Google, Nike, Alex, Citizen. So several companies are available in the market. All the physiological parameters are monitoring by using these sensors, and they are very much important for fitness monitoring application. The third one is for rehabilitation. So these types of sensors are important for, like, people undergo like paralysis, some stroke happen, and the person cannot, the nerve activity or the muscle activity of the person is not good. Then for those peoples, we need like a continuous treatment or a continuous monitoring, and periodically we have to apply some electric stimulation to improve the nerve or muscle activities. So for that purpose, we can use some wearable patches and periodically that patch can apply electrical stimulation, and we can measure how the muscle or the nerves are responding towards these responses. So no need every time the 24 hours, no need of doctors monitoring. We can attach a patch, and the doctor can, nowadays cloud devices are available in the market. So wherever the physician is, they can monitor the status of the patient. So now let us think about why we need a wearable electrochemical sensor. Earlier I mentioned about several POCT devices. So we have to collect blood sample or any other biofluids. Basically, we have to prick our finger, or by using a needle, we have to collect blood samples for further analysis. But think about some diabetes patient, and they are a very old person. They have to have the analysis like periodically, maybe before food and after food. But some people are having insulin daily 3 times or 4 times, and periodically they have to monitor their blood glucose level. So for them, always pricking their finger is really difficult, and it is painful in nature. And for if we consider a newborn baby, so it's really difficult, and the blood sampling is a tedious process. So for that application, we can, apart from blood samples or blood serum samples, we can think about other biofluids like sweat, saliva, or interstitial fluids, and using non-invasive way or like a semi-invasive way, we can access these biofluids and have electrochemical detection. So then I said about if it is a wearable sensors, especially for a chemical monitoring application, we need to think about the mechanical properties of those sensors. Mechanical properties in terms of flexibility, stretchability, and self-healing ability. So why we need these physical properties? So the main property is, so if we are thinking about sweat collection or interstitial fluid collection, so we need to have a stable and conformal contact with the human skin. Then only we can periodically we can collect the samples, and we can do the analysis. But if you think our human body is not very flat, our human body is three-dimensional in nature. There are so many curves, and it's curvy linear in nature. So consider if we prepare a rigid sensor, and if we are trying to attach on our human body, we have to apply some pressure. So during that time, sometimes the sensor can damage, it can break, and we cannot have 100% conformability, the skin conformability using rigid sensors. So if the sensor is flexible in nature, we can easily, the sensors can easily adapt with the three-dimensional skin or the epidermis surface. And in another one thing is the stretchability. So no matter how much flexible your sensor is, sometimes we are attaching the sensor with the human body, and the person is doing all the regular activities. They do exercise activity, or so during these all normal activities, our muscles, there will be so much movements, or muscle or skin can stretch sometimes. Sometimes it undergoes a twisting movement. So all these movements can create strain on our sensation. So what will happen? Sometimes there will be some micro crack formation on the sensors, and so we have to develop the sensors with the stretchable property. And another one is a self-healing property. even if some cracks are happening, if the electrodes or the sensor have the self-healing property, it can heal by itself, and we can continuously use these sensors. We will see different types of these devices. So as I mentioned earlier, the wearable electrochemical analysis can be done from different biofluids like a sweat, interstitial fluid, tear, or saliva. First one is sweat. From the sweat, we can have both clinical analysis and fitness monitoring. So as you can see, several analytes of interest are present in the sweat sample. So we can detect lactate, glucose, uric acid, ascorbic acid, ethanol, and these ions and cortisol, etc. So lactate basically it is a biomarker for tissue oxygenation. Glucose, all we know, it is related with the diabetes. And sodium, it is related with the hydration level. We know hydration, it's like a very serious condition, health condition. If more water is losing from our human body, it will leads to the death of the person. Then chloride, it is important for monitoring cystic fibrosis, and pH, uric acid, cortisol, everything is important for, it has its clinical application. So basically, sweat is a product which is produced by the thermoregulation. So which is a product of thermoregulation of our human body. So basically, it consists of a dermal duct and a secretory coil. So whenever there is a change in temperature, or there is a change in emotional level, the secretory coil activates and it produces sweat, and the duct carries sweat to the upper part of the epidermis. And from there, if we are attaching our wearable sensor, we can have this analyte of interest. So for as I said, it is a thermoregulation process. But what will happen, for how we can use sweat for a clinical diagnosis? For a clinical diagnosis, we are dealing with the patients, and we cannot ask the person whenever we need sweat. We can't ask the person to ride a bike or do some exercise activities. So it is very difficult. For a fitness monitoring application, no problem. We can do the exercise activity, and immediately within like some minutes, we can collect sweat samples and do the analysis. But for clinical application, we need to depend on a technique called iontophoresis. So in iontophoresis, many of you know, which involves a transfer of ions with the help of direct electric current. So we have to apply 2 electrodes on the epidermis, and we have to apply some direct DC current. Especially in hospitals, they are using for administrating drug samples. So the medicines will be placed under the electrodes in the form of some gels, and with the application of current, what will happen? So the medicines, sometimes it will be in the form of ions, or the application of electric potential, it polarizes the medicines, and based on the polarity of the electrode. So for example, negatively charged ions repel from the negatively charged electrode. So due to the difference in polarity, we can administrate, or we can deliver different drug samples. So for our sweat stimulation application, we have to use a drug called pilocarpine. Okay, so we have to develop some gel containing this pilocarpine drug, and by use of 2 electrodes, let's say for example anode and cathode, we can deliver this pilocarpine. So when it reaches these secretory coils, automatically the sweat stimulation happens, and we can access sweat at the epidermis. And another one technique is available, like reverse iontophoresis. which is not related with the sweat, but related with the interstitial fluid. So only one difference here in endophoresis, we are using pilocarpine drug and applying potential. But here we are in the absence of pilocarpine, we are applying the electric current to the human skin. So here it depends on the electro-osmotic flow of interstitial fluid. So initially, what will happen? The anions are present in our interstitial fluid, like a chloride ion. It will move towards anode because the anode is positively charged, and the cations like a sodium, it moves towards cathode. So this movement of ions causes electro-osmotic flow of interstitial fluid, and along with the fluid, the neutral molecules and several other analyte of interest also move towards the skin surface, and we can have the detection. So now think about major platforms for preparing these flexible sensors. So either we can use cheap or inexpensive plastic substrates, like our general OHP sheet itself we can use as a substrate. The only one condition, we have to use some thin substrates which is really flexible in nature. So these are all the plastic substrates. Some of them are it can temperature stable. So Kapton we can apply high temperature. So easily we can coat any electrodes on top of the Kapton substrate and use it. And another one is a textile substrate. So textile substrate opens an advantage, no need of any external substrate. Everybody is wearing some t-shirt or some clothes. So this fabric itself we can use as a platform for our wearable sensors. So we, either it can be from wool, like animal source, or a plant source. So any different source, no problem. We can directly print the 3 electrodes on top of the textile substrate. And the only one condition, these fabric substrates should have conformal contact with the human skin. For example, t-shirts. So if we are using a tight t-shirt, so we can easily use that as a substrate for wearable application. And coming to the fabrication protocol, we have several techniques. One is screen printing, and many advanced techniques like lithographic deposition, solution processing, transfer printing, or nanomolding techniques. But out of these techniques, screen printing technique is a very inexpensive technique. And so these are some of the examples of screen printed electrodes, and these are some of the lithographically derived devices. So the one advantage of lithographic devices are we can have very extremely thin coatings, and we can control the resolution even micron size or less than micron size. But if we consider screen printed like 50 or 100 micron size, above 50 or 100 micron only we can have the electrochemical sensors. So this is the schematically explaining how we can prepare screen printed electrochemical sensors. So initially, earlier I mentioned we have to use AgCl ink and carbon ink. So we have to basically prepare a stencil or a screen that has the design of the electrochemical sensor, and we have to apply the ink on the stencil, and using a squeegee we can move the ink on top of the stencil, and this leads to the printing of that ink at the on the underlying substrate. So after curing the first print, then we can choose the carbon stencil. The 2 different stencil we have to use. One for printing AgCl ink. So all the contacts will be printed initially. Then we have to print the working and counter electrode. For that purpose, carbon ink we can use. So finally, we will get an ideal 3-electrode system. And one more step, we have to define the area of the electrode. So for all electrochemical sensing application, we have to fix the area of the working electrode. So for that purpose, on top of this electrode, we have to print some dielectric ink. Mostly they are curing by UV, just exposed to UV light, and it will immediately start the polymerization. And within some less than 5 seconds is sufficient for curing up that this insulator ink. And we can use this screen printing method for printing electrochemical sensors on a rigid substrate like alumina, or as I mentioned, textile substrate we can use. And another one very good example is a tattoo paper. Just nowadays, young generation, they are very much interested in applying temporary tattoo on the human skin. So these are the tattoo paper. Tattoo paper is nothing but it is a paper substrate which have an upper layer of a water soluble coating. So easily we can coat or print our electrodes on top of the tattoo paper, and we can transfer easily on top of the epidermis. So any design we can, depends on our application, we can design our electrodes and prepare a tattoo and attach on the skin surface and do the analysis. Even on top of a bandage, it's also we can use, and we can monitor the wound healing process, the monitoring of wound healing. And not only for healthcare application, it has several other application like forensic application also. So now I'm going to show some examples of these tattoo-based and other different sensors. So this is one of the epidermal tattoo developed in our lab, and we used this for sodium monitoring application. As I mentioned earlier, the monitoring sodium is really important for persons with a cystic fibrosis. So this cystic fibrosis affects the normal tubes or ducts of our human body, especially where it produces mucus, sweat, and digestive fluids. So when these passage ways are plugged up, what will happen? It affects the release of these fluids, and the fluids become thicky and very sticky. So this will increase the amount of sodium in our human body, both in sweat as well as in the human blood. So this disease especially affecting by the young inborn babies. And for fitness monitoring application also, it is really important. So we can easily access sweat. So here one person is wearing this tattoo, and this is a controlled PCB circuit board. So which can these ion sensors are basically potentiometric sensors. All other metabolites are depending on enzymatic amperometric detection. But for ions, we cannot use enzymes for any other antigen or antibodies. So easily by using potentiometric sensors, we can detect. So we have to modify the working electrode of the tattoo, and which consists of some specific ion selective molecule. So some membrane which consists of ion selective molecules and some charged carriers. And whenever we are changing the concentration, so the sensors can easily show the exact concentration. So I'm not going to the details of the sensor. So another one tattoo sensor here I'm showing, which is using for lactate monitoring application. So lactate, which is basically a product of anaerobic metabolism of anaerobic metabolism. So usually, if sufficient oxygen is present in our human body and in our cells and tissues, the metabolism follows a particular route. But whenever if we are doing too much exercise activity, and then the body lacks sufficient oxygen. So at that time, our body produces more and more of lactate. So lactate is a biomarker for tissue oxygenation. The insufficient tissue oxygenation we can detect by using monitoring lactate from our sweat samples. So another one tattoo developed, and when the person is doing the stationary biking, we can collect the sweat samples and directly do the lactate analysis. You can see in this figure. Initially, some 10 minutes there was not much perspiration, and when the perspiration starts, we can see the current signals proportional to the lactate concentration. So when we increase the exercise intensity furthermore, we will get more and more lactate concentration. So what will happen if if our tissues don't have sufficient oxygen? So it will leads to the muscle cramping. So for our bloods, the muscle cramping is a serious issue. So if the cramping is happening when they are doing exercise activity, it will damage our tissues or our muscles. So by monitoring lactate from the sweat, we can avoid the possibility of muscle cramping. So this is another one example of potentiometric detection of ammonium from the sweat sample. So monitoring ammonium also important for monitoring hepatic disorders and liver malfunctions. So I'm coming to our recently developed wearable microfluidic sensor. So earlier I showed 3 different tattoo-based sensors. So one main problem associated with these sensors are they are directly we are attaching on the human body. So one problem, our sweat or human body consists of several dust and other impurities. So sometimes over the if we are using for a long-term application, the surface of the electrode will undergo fouling. So some proteins or dust it will go into the some enzymes are there, it will damage the enzyme activity, and further we can't use the sensors. And another one problem, if we are doing more and more exercise activity, sometime there will be accumulation of sweat on the electrode surface, or sometimes dilution of sweat happens. So we need to avoid these problems, these direct attachment of electrochemical sensors on the human body. So for that purpose, we can use these microfluidic channels, which can be embedded on the screen printed electrodes. So this is the electrochemical sensor prepared on a polyimide substrate or the Kapton substrate. So initially, our aim was, so earlier I showed a sensor is monitoring only a single analyte, but here our intention was monitoring multiple analytes simultaneously using a single patch. So we developed these electrode design, which consists of 4 different working electrodes. So here we follow the 2 electrode system, only not 3 electrode. We can use 2 electrode also. That means the reference electrode we can use for both purpose, like reference and counter electrode. So ideally we need 3 electrode system, but for practical application we can use 2 electrode system also. So and we developed the microfluidic channels. So usually in worldwide, microfluidic channels are developing using advanced lithographic techniques, and we definitely need clean room facilities. But here we followed another one technique, like the same screen printing technique. We printed carbon ink on a flexible substrate, and on top of the pattern, we printed a silicon rubber, which is the flexible polymer. Silicon rubber is nothing but all PDMS, the commercially the PDMS, which is very well elastomeric substrate. We can have mold transformation. So after curing this silicon rubber, we can have these microfluidic channels, and these channels we can attach on top of our screen printed electrodes. And as you can see, there is 4 different sensing sensing areas, and all 4 areas are covering with these microfluidic channels. So whenever the person undergo, so here we attach these developed sensor on upper back region, and when the person undergo a stationary biking, here this is the inlet. So from the inlet, the sweat enters, and it allows a real-time flow of sweat over the transducer. That means over the sensor surface. So there is no direct contact of electrodes with the skin, and we can have a real-time. So we can avoid the dilution, because every time when the sweat reaches at the inlet, it flows over the electrode surface, and there is no accumulation or dilution of the sweat sample. And here we used I mentioned earlier, for 4 different analytes we have chosen. One is lactate, another one is sodium, potassium, and pH of sweat. So all these analytes are really important. So our main challenge is how these lactate sensor is amperometric detection, and other ions are potentiometric detection. So when we apply potential for amperometric measurement, definitely the current flows through the non-selective electrodes for potentiometric detection. So there will be current interference from the amperometric sensor. So in order to avoid, so initially we conducted our these experiments using our commercial instruments. So we observed the current flow. We cannot detect any ions from the solution. The open circuit potential was very large because of the amperometric interference. So we prepared the circuit board in such a way. We made all these potentiometric sensors like floating in nature. So for electronics people, they can easily understand. We are not grounded these potentiometric sensors, and only the amperometric sensors are grounded. So then we used for wearable application. So this one subject undergo cycling, and we can have 4 different, the detection of 4 different analytes in a real-time fashion. Like 30 minutes or 1 hour, we can continuously do the analysis. And using this microfluidic sensor, we can effectively avoid the surface fouling of electrodes. So we can use this sensor like 1 day or 2 days. If there is no fouling, still the sensor is active, and we can use these sensors for continuous monitoring application. And now I am going to show another one flexible sensor we developed, and we used for both chemical monitoring as well as the electrophysiological monitoring. And that means if we monitor only chemical parameters, it won't give the complete parameters, the complete idea of the underlying physiology of the human body. So definitely we need to monitor both the parameters simultaneously. So for that purpose, we have screen printed sensors which have ECG monitoring electrodes and the lactate. This center 3 electrodes are for lactate monitoring. So this PCB which can collect both the signals simultaneously. We have to attach this sensor at the chest V1, V2 region, and we can have the real-time detection. And earlier, now I'm coming to the interstitial fluid. I mentioned we can effectively harvest sweat and do the electrochemical analysis. And another one biofluid is interstitial fluid. So earlier I mentioned the reverse endophoresis. So using this technique, we can harvest interstitial fluid by electro-osmotic flow. So then we can use. So here it is screen printed carbon as well as the endophoretic electrodes. That means AgCl electrode. So whenever the fluid reaches at the working electrode, it can detect glucose with high precision and the high selectivity. So due to a short of time, I'm moving forward. So another one method is we can use some micro needles to detect to monitor interstitial fluid. The another one here I mentioned the reverse endophoretic technique. We can extract the interstitial fluid. So here another one which is a semi-invasive technique. We can prepare the hollow micro needles using injection molding techniques. But for electrochemistry for application, we need 3 electrodes. So we can choose platinum wire for working and counter electrode, and we can insert a silver wire for reference electrode. And you can see this is a very tiny device. You can see at the fingertip. And the working electrode we have to modify with this sensor we used for alcohol monitoring application. So for that purpose, we modified the electrode, working electrode with the alcohol oxidase enzyme. And in order to improve the performance and avoid interference, we coated some polymer, we retropolymerized some polymer and naphthalene also. So as you can see, the microneedle sensor can detect alcohol it shows like a wide range of detection, and it shows a very good selectivity and stability. And finally, we used the sensor for like ex vivo analysis. We prepared artificial interstitial fluid, and we can't use microneedle directly to our human skin because we don't have such a permission because of the safety issue. So we choose mice skin samples, and you can see the microneedles can easily penetrate the mice skin and can access the interstitial fluid. So when we vary the concentration of alcohol, we can easily monitor the current response to the alcohol level. And another one biofluid is saliva. So saliva also consists of several biomarkers. And if you think about the platform, many older peoples and the athletes are using mouth guard. So mouth guard is a very good platform for integrating these flexible sensors. So this is one example of ambarometric lactate detection. But we need to take very care about the biocompatibility because we are directly putting these mouth guard with our human in our mouth. So we have to choose the molecules like guard materials. Everything should be safe, like it should be biocompatible. Then only we can use these mouth guard based sensors for continuous monitoring of saliva. So this is another one example of uric acid sensor based on these are all are I'm showing like prototypes of current devices not currently available in the market. So many of the sensors are many companies are working on the commercialization process. But remaining all I am showing the translational research. the proof of concept many research groups are working on, especially the University of California San Diego campus. They are very good. The Dr. Joseph Wang group, they are very good in developing these wearable prototypes. So I'm due to lack of time I'm moving towards stretchable sensor. So earlier I mentioned, no matter how much flexible our sensor is, if we flex like 180-degree, if we bend, then it will undergo some kind of damage. If some micro cracks appearing on the electrodes, it will adversely affect the conductivity and all the performance. So there are mainly two different approaches are available to make the electrodes more stretchable in nature. One is a random composite route. So in this method, we are using elastomeric stretch enduring binders. So several thermoplastic polymers are available, and some block polymers of polystyrene. So these class of polymers are very good, stretchable in nature, and they can endure high level of strain and protect the devices. So these are some of the examples of the binders. And another one is deterministic approach. That means design induced stretchability. So we can have these different designs. One is a wavy shaped platform, and another one is a ribbon shaped platform. Then the third one is a serpentine shaped platform. So how these designs increase the stretchability of electrochemical devices. So whenever we stretch, so I will show one of our work. So we developed some island bridge devices using, we combined two different techniques. One is a lithographic technique and another one is a screen printing technique. So initially we developed this pattern. So here these yellow color region are active sensor area, and which is interconnected with, as you can see, serpentine shaped bridges. So these are contact paths, and when we stretch these sensors, what will happen? Our consider a spring. Spring is like a three-dimensional nature, but serpentine is like a two-dimensional spring. If we stretch a spring, it won't easily break. Or we can stretch to a particular distance. So same thing happen. Only these all the strain goes to the these serpentine patterns, and unwinding of these serpentine patterns protects our electrochemical sensors. So here this is the image of showing we applied continuously 50 pinches, and later we stretched the device, but the device showed robust performance. So here it is a we characterized the stretchability in terms of measuring the resistance. We apply different level of stretching, and we simultaneously we monitored the resistance change. As you can see, there is very little change in conductivity, and electrochemically also we characterized. So these sensors we can stretch not only uniaxially, we can stretch in both the direction, and we applied these sensors for monitoring DNT and glucose, lactate. Several analytes we tried, and the sensor works very good. And finally, we did on-body analysis, and we can able to detect a lactate, the real-time lactate level easily from the sweat samples. Sir, can I have like 10 more minutes?
Sure, sir, you can continue.
Yeah. Yes. So the third parameter earlier I said, the self-healing sensors. So the first one is flexible, and the second property stretchability. So no matter how much stretchable our sensors are, if our muscles can stretch a lot, and during this in exercise activities, there will be lots of damage. It can happen on the electrochemical sensor. So if some damage is occurring, some micro cracks are happening, if the system can heal that damages, and then we can use those sensors continuously. So there are mainly two different approaches. One is capsules loaded with self-healing agents. Okay, when we are preparing the electrochemical sensor, that means the electrode, we have to load some capsules which consists of some organic solvents. So basically what will happen, consider this is the electrode, and when we are damaging the electrode, if some damage is happening, or externally we can cut, using a scissor or knife, we can damage externally. What will happen, these capsules are loaded with the healing agents. So that capsule release these solvents, and locally it will undergo reorientation of binder and conductive filler particles. Okay. Basically, we have to choose these healing agents. So which binder we used, the compatible solvent we have to use, and we have to make sure these capsules are not affecting the electrical conductivity or the electrochemical property of the electrodes. So we have to use very low concentration of these capsules, and it should be in micro level. Then only we can have a homogeneous distribution of these capsules, and these releasing of agents can self-heal the sensor by itself. But one problem with this approach is we cannot have multiple healing at the same location. So once we damage some of the capsules, what will happen? Then there is no more capsules are available with the healing agents. So for that purpose, we can use another one approach. We can, while preparing the electrodes, we can have dynamic chemical bonds. So these chemical bonds, we can form a reversible bond formation. And whenever some damage occurs, immediately, like instantaneously, the reversible bond formation happens, and it leads to the self-healing process. And the chemical bonding, there are two different types. One is a non-covalent interaction, and another one is a covalent interaction. So non-covalent interaction can be Faust-Gust, hydrogen bonding, or hydrophobic, or metal coordination. In the case of covalent interaction, these are the possibilities. Using this type of approach, we can have multiple healing at the same location. And another one approach, we recently used this approach for developing magnetically self-healing electrochemical devices. So what we used a permanent magnet particles, which is Nd2 neodymium ferrite based particles. So we, while preparing the electrochemical sensors or the electrodes, we mix these magnetic particles also, and when we screen print, we align the polarity of the magnet using another permanent magnet. So if we are not aligning, then we cannot have the magnetic property. During the printing itself, we have to align in one direction. Then if you cut the electrode into two, each pieces shows the magnetic attraction. So when we damage, if you damage completely, like not only electrode, the electrode and the substrate also, if you are cutting with a scissor, but like from 1 mm distance itself, the self-healing can happen due to the magnetic attraction of two different traces. So here you can see we used for H2O2 sensing as well as the CO2 plus sensing application. During the sensing, we damage, we basically using a scissor and cut the electrodes multiple times, but still we can perform the electrochemical analysis. Simultaneously, we did copper sensor, copper analysis. So using this sensor, we can, even if the damage happens completely, like electrodes and the substrate, everything, if we completely, if we damage also, we can instantaneously, we can regenerate the electrical conductivity and do the analysis. And finally, I'm coming to the our recent work related with the smartphone based electrochemical sensor. So all the everybody have a smartphone with us nowadays. So for a diabetes monitoring application, as I mentioned, whenever people are traveling, they used to keep their glucometer with them, so that periodically they can track their glucose level. So what will happen? Sometimes if the patient forget to take the glucometer, then it is very difficult. And what if we are attaching our electrochemical sensor with the smartphone itself, then no problem. Every time we are carrying our smartphone with us, and we can do the analysis whenever we require. For this purpose, we developed a 3D printed case, because everybody is using case for their phone. So this is the 3D printed case, and this is the place for the electrochemical sensor. You can see the 3 electrode system, and we developed a PCB for controlling the sensor, and which can be controlled by, powered by the phone battery itself. And one more thing, one added advantage here is the sensor can be, we can use multiple times. So every time no need to remove this flexible or the disposable sensor, we can remove the active layer. So here we prepared the active layer in form of pellets. So you can see a 3D printed pen is available. So it filled with several pellets. So inside the pellet, we loaded the active materials. And one more thing, under the working electrode, we attached a small, tiny magnets. So what is the purpose? And while preparing this active material, we mixed with some magnetic powders. So whenever we are ejecting one pellet to the area, so no need to align due to the presence of underlying magnet. So this glucose pellet, the active layer, directly it will go and attach on the transducer, and then we can do the analysis. So as you can see here, so we developed a Android-based app, and when we expose different concentration of glucose to the electrochemical sensor, when we put 60, we got 61 milligram per dl. When we apply 100, we got 103. Very mild variation. For 190, it showed 194 milligram per dl. So using this prototype, we proved that we can prepare smartphone-based electrochemical devices, and wherever we are, if we are doing some exercise activity, or if we are doing, if we are traveling, or wherever we are, just open the app and load one active membrane on the electrode surface, and simply we can do the electrochemical analysis. So with that, I'm winding up today's talk. So I'm happy to answer any questions from the audience.
Thank you so much, sir. It was indeed an insightful session containing a lot of information, and you covered all the aspects related to the topic with well-detailed PPTs. We've received some queries regarding the session. I hope you don't mind answering them.
Yeah, no problem.
Others may kindly raise your queries in the chat box, or you may unmute yourself and ask. So, sir, shall I read it out for you?
Yeah, yes.
Okay, sir. So the first question is, kindly inform about Indian scenario of wearable devices especially whether they are manufacturing anywhere in India, including CE/CRI?
Can you the first part of the question? What's that?
Kindly inform about Indian scenario of wearable devices.
Oh, Indian scenario. Okay.
Yes.
Actually, in India, very few labs are working on wearable electrochemical sensors. Several labs are working on electrochemical sensors based on screen printed electrodes. Several labs are working, but only very few labs like IIT Kanpur, IIC Bangalore, and then of course SIPRI. So we are working on like preparing flexible and stretchable electrochemical sensor and integrate microfluidic channels and do the actual wearable sensing. And for the commercially, right now, earlier I mentioned only the Abbott company, they developed the wearable glucometer. And several these flexible sensors are like at the lab level, or the translation process is going on. Yeah.
Okay, sir. So the next question is, what kind of help or support need to be extended by organizations such as IEEE, TSI, Digital Health India Association for translating the innovative sensor designs to medical products and associated services in India?
Yeah, for one, if we are considering wearable application, so I mentioned the wearability in terms of sensor only. That means the screen printed electrodes we are fabricating in a like flexible and stretchable form, but not the controlled circuit board. So in order to have an ideal wearable sensor, we need to develop these circuit board also like flexible in a manner. So for that purpose, some works are going on like instead of using the rigid PCB board, we can use the Kapton substrate. I mentioned Kapton can the high temperature it can withstand like above 700 or 800 degrees Celsius. So some groups are working in a broad. They are using a Kapton substrate and they are preparing the controlled PCB. And but in India, I don't think any groups are working on like really flexible circuit board. So if we are using those circuit board, and one more another challenge, if one person is working in electrochemical sensor, they are not thinking about integrating with the circuit board and like integrating with any Android app or something. So in India, we need to concentrate on more interdisciplinary work. So we need to have one chemist, one physicist, or like IEEE person. So then only these interdisciplinary research only can solve all these issues. And another one, earlier I mentioned, we need to avoid the sensor to sensor interference. And when we apply the current to the human skin, it should not damage. So sometimes some people's skin is very resistive in nature. So at that time, the current flow will be more. So sometimes it damage the skin surface, especially for iontophoresis application. So I have experience a little bit in iontophoresis. Many times, if the fluid content is for the gel ionic strength is not enough, definitely it damage our skin surface. So these problems we need to address.
Thank you, sir. The next question is, can the sweat analysis results vary from a person having normal sweating to abnormally excessive sweating?
Yeah, yes, that also another one challenge regarding sweat sensing application. So even it varies with the ethnicity, like people from Asia, and if you are considering from Europe or Africa. So the nature of thermoregulation of people is different, and it varies from person to person and varies from male to female. So we need to have some in situ calibration, like a 1 point or a single point calibration we need to have. Then only we can, because the initial calibration parameters that will come from one particular person, but that calibration parameter we can't apply for everybody because of the difference in biomarker secretion. So for that purpose, we need to add a single point calibration. So first to do the calibration, then do the analysis. So then we can solve these issues.
Thank you, sir. The next question is, do these wearable sensors lead to any allergies or irritation to the skin surface due to excessive usage?
Well, then we have to, while fabricating the electrodes and the other, we need to take care when choosing the substrate or electrode or other components. Everything should be biocompatible in nature. And sometimes what will happen if we are using some mediators, for example, for glucose analysis, not only the enzyme we are using some other mediators also. Those are some organic molecules. So that itself can sometimes, when the person sweating a lot, the mediator leaches from the electrode surface, and it can have cause some skin irritation. So for that application, definitely we cannot attach the electrodes directly on the human body, and we need microfluidic channels, so we can avoid direct fluid contact. And from the outlet, there is a chance of from the outlet it will come to the skin surface. So there we need to have some fluid absorbing pads, so that completely we can protect our skin from these irritations or toxic behaviors.
The next question is, both sensors are very costly. Can we develop a simple glucose watch?
Glucose, yes, I think several companies are now working on developing watch kind of glucometers. Yes, it's definitely it's possible, but currently everything concentrating on harvesting interstitial fluid from the skin surface and to do the analysis. But from the, of course, there are so many challenges associated with the sweat-based sensors in terms of the reproducible sweat secretion rate. So definitely we can, in interstitial fluid based glucometers like a gluco watch can be easily fabricated.
Can I ask a question? This is Dr. Kulkarni from Mumbai.
Yeah, yes, sir.
This is in continuation with what we are discussing.
Okay.
See, this is not a very big rocket science. Just to have one glucose detector attached to a base of a watch, and either it can give a low value or a high value. It need not give the digital value also.
Yeah,
Because Abbott has got 14 days sensor, cost 5,000 rupees for the sensor,
yeah,
and 5,000 rupees for the reader. The same thing
yes.
was available at the 45% of the cost 2 months back.
Yeah,
Means
yeah,
when we are talking of India, and when you
yeah,
are collaborating with so many institutes like IIT and other things, I think this should not be a very, very big issue. I mean, India, there are more than now 16 crore diabetics.
yeah,
So
yes.
if you think properly, and if you combine, and a wonderful idea will be combining the lactate sensor with the glucose sensor, because lactate will give idea of the muscle and glucose will give the idea of glucose metabolism. And if you combine it again with a smartwatch like Fitbit, then it will be a complete, almost like a digital twin of that person. This is a
Yeah,
thing which you can go ahead. And if you need any help, I am texting my email in the chat box. You can be in touch with me. I am working on technology in medicine for almost last 12 years. Thank you very
yeah.
much.
Yes, sir. Yes, sir. Thank you, sir.
Thank you, sir. The last question for the session is, what is the main roadblock of commercialization of wearable sensors in India?
So the main challenges, of course, the sensor should be completely reproducible in nature. So we have problems associated with the functionalization of the sensor sensing surface, because right now in the lab level we are functionalizing like glucose or enzyme, whatever the surface functionalization we are using with our hand. So definitely we need to have some instrument for precisely monitoring. We need to control the active layer in terms of either volume or in terms of like a weight. Then the reproducibility or the repeatability of the sensor, it won't be a problem, because in the commercially available glucometer, they are telling the error percentage of sometimes 15 to 20%. So that much error is available in the commercial glucometers itself. So we need to have specific attention for making the electrodes or the strips like more reproducible or repeatable in nature. And then we need to have the controlled glucometers. I think if we are combining these type of platforms like digital healthcare association, so if we are combinedly working, definitely the problem in India, earlier I mentioned, some people are working on the basic research, and some are in translational research, and many of them are not focusing on the complete product development. So we are having a good collaboration. Definitely we can, earlier sir said like low or high, no need of like a perfect meter, like a low or high, or those things are really easily we can fabricate by if we are using two amperometric sensor. Yes, we need to take care the current flow is not the current interference from sensor to sensor. So those things we need to take care. Maybe the electrical engineering engineers, they know very well how we can do parallel amperometric detection simultaneously. So that we can easily solve these issues and go for commercialization.
Thank you, sir. I believe that's the end of our query session. Thank you so much, sir, for clearing our doubts and enlightening us more on the topic. Now, as a token of love and appreciation for the impeccable session you've delivered, we'd like to share our gratitude by presenting a virtual memento. Manoj, sir.
Please accept our virtual memento here, Dr. Vivocity.
Yeah, thank you, sir. Yeah, I can see. Should I do anything?
Kindly accept this, which we consider as a representation of our boundless gratitude and acknowledgement for your valuable time and effort.
Thank you so much.
Thank you, sir. Now I invite respected Dr. Uma Nambiar ma'am, Chairman, Digital Health India Association, to deliver the vote of thanks.
I thank you so much for inviting me here. And I must congratulate Dr. Vinu Mohan for such a wonderful talk, because I think, you know, in the series of talks that I have been part of related to telemedicine and things that are making an impact, this has been one of the most knowledgeable, and so much of amount of knowledge has been packed in one hour. I'm sure everybody here understands and realizes that I think 1 hour was too little for a topic such as this, but thank you, Dr. Mohan, for condensing it so well. And the proof of the pudding lies in eating. So the participants who were there right from the beginning, and I was observing, not even one person dropped out. And that in itself is commendable, and that's a testament to the fact that everybody was so glued into your lecture. So thank you very much for bringing to the fore one of the topics that has gained more importance now in the light of the pandemic and the kind of explosion that we are seeing for remote evaluation and remote monitoring, as well as remote care itself. So I think, this is the future technology in healthcare. I mean, healthcare is completely getting transformed now to digital, to more remote, to more personalized. And when that happens, I think wearable sensors are going to play a very, very important role. I mean, when we talk of personalized medicine, I think one of the starting steps for personalized medicine will be when all of us will have sensors of various kinds, which are very individualized and which take care of us. So I think you have highlighted something so important.
I think ma'am have got a network issue.
Yeah, a little bit connection problem.
Okay. I kindly request all the participants to fill the feedback form in the chat box. Thank you. Thus, we have arrived at the end of today's session. We are very grateful for having you all today and for your kind cooperation. I'm pretty sure the webinar has flashed insights to your mind. Once again, on behalf of IEEE MBS Kerala chapter and all the collaborators, thank you from the bottom of our hearts. Thank you, sir.
Thank you. Thank you so much.
This is Amina signing off. The meeting is officially closed.
Hi, sorry, I got disconnected from another network.
Yeah.
Yeah, so I'm so sorry. Yeah. So as thank you so much for, I really, technology really, bothers you. I had an incoming call on this. So Dr. Manoj, yes, thank you very much for, selecting this topic and selecting such a wonderful speaker. And Dr. Ram Kumar Menon, my fellow neurosurgeon and fellow TSI member, for introducing the speaker and doing such a wonderful job today.
Thanks,
Dr.
ma'am.
Korus Varghese, former chair of the IEEE, and almost everyone who is here today, the members of IEEE, all the TSI member, DH India members, the student volunteers. And once again, I want to thank the audience, because without their sustained presence here, this lecture could not have been, could not have reached its proper conclusion. So you've really done justice to the session. So thank you all once again. Thank you very much.
Thank you for your valuable words, ma'am. We very much appreciate it. I hope you all have filled the feedback form. Once again, thank you from the bottom of our hearts. The meeting is officially closed.
नमस्कार, मी कॅप्टन वैभव दळवी. वयाच्या 50च्या वळणावर
Hello.